For my inquiry, I investigated how different types of artificial light affect the photosynthetic activity of isolated chloroplasts. For inquiry 4, I investigated how different types of artificial light affect the photosynthetic activity of isolated chloroplasts. Light is necessary in order for energy transfer to take place in plants. In 1937, Robert Hill actually found that CO2 was not required in order for photosynthesis to take place. He demonstrated that oxygen was reduced during the light-dependent steps of photosynthesis in the absence of CO2, so it only really required light. Most plants grow with the sun as a light source, but photosynthesis can take place under artificial light as well. The main differences between light sources are the intensity and the wavelengths that the sources emit. The intensity is the brightness or the number of photons or units of light absorbed by a unit of area. Plants primarily absorb blue, red, and yellow wavelengths. 4 light bulbs used in this experiment: high intensity discharge or HID bulbs, light emitting diodes or LED, fluorescent, and incandescent. The sun was not used as a light source because of how difficult it would be to measure it. Photosynthesis guides the transformation of light energy to chemical energy in the chloroplast of plants. Within the chloroplast, reactions that depend on light happen in the thylakoid membranes. Here, carbon dioxide and water are converted to carbohydrates and ATP, which is food and energy for the plant. The transfer of electrons happens here in two photosynthetic systems. Water molecules are split apart in photosystem 2 to give off oxygen and an electron. The electron is transferred to NADP+ through photosystem 1, producing NADPH. ATP is produced after photosystem 2 produces an electron gradient across the thylakoid membrane. Robert Hill used an artificial electron acceptor in his experiments in the absence of CO2. The artificial electron acceptor that was used for my experiment is DPIP. As DPIP accepts electrons, it becomes lighter in color, changing from a deep blue to clear. First, 10.5 grams of spinach leaves were weighed and blended. SPP buffer was added in the blender to take away any unwanted cell material and to separate the chloroplast from the other cell products without damaging the chloroplast, which is what I needed. This mixture was filtered and centrifuged at 4 degrees and at a force of 1500 times gravity. Since centrifugation releases degradative enzymes, which are not active at cold temperatures, it is important to keep everything very cold. Centrifugation caused a pellet to form at the bottom of the centrifuge tube. This is the chloroplast. The rest of the solution was discarded, and it was centrifuged a couple more times with SPP buffer to isolate the chloroplast further. After I had the chloroplast suspension, I covered test tubes with foil and prepared them with SPP, chloroplast suspension, and DPIP. This is what I exposed to the different light bulbs in 30-second increments. The solution started blue, but as DPIP was reduced, the solution turned back to light green. So I was able to measure the absorbency of the solution over time using a spectrophotometer. The blank I used did not have DPIP in it, so I could tell the reaction reached completion when the absorbency reached 0 and turned back to green. Fresh spinach was used every time I started the lab. It was crucial for the DPIP and the test tubes to be covered in aluminum foil so no light-dependent electron transfers could take place. DPIP and SPP are skin and eye irritants, so I used a lab coat and gloves in the biochem lab. I labeled everything I used and disposed of solutions in waste bins under the hood after I finished my lab. These are the means of my results. As you can see, the chloroplast solution that was exposed to the fluorescent bulb reacted at a faster rate than the others, between 30 seconds and 4 minutes and 30 seconds, while the others seemed to react at a similar rate. This surprised me because I thought chloroplast exposed to incandescent bulbs would photosynthesize at a slower rate than LED and HID. Not all starting rates were the same because DIP is light sensitive. When I transferred the chloroplast solutions to cuvettes, the reaction could have started. I tried to minimize this as much as possible by covering the cuvettes before starting. With a confidence interval of 95%, my t-tests revealed that the differences between the rates of photosynthesis were not significant between LED, HID, and incandescent light sources. Chloroplasts exposed to fluorescent bulbs are significantly different than the other three throughout the reaction, though the total time of the reactions did not differ by much. Two other fluorescent tables are not shown here because the results are similar. Errors could have happened because I did not grow my own spinach and because there were different starting rates when the chloroplasts were reacting. Fresh spinach was bought every day in order to minimize possible error from using old leaves, but since I brought the spinach from HEB, I had a limited control over how fresh the spinach was or if there were any pesticides or herbicides used on or around the leaves. From my data, I can conclude that fluorescent bulbs enable chloroplasts to photosynthesize at a faster rate than incandescent LED and HID bulbs, but the overall reaction time between all four is not significantly different. I only did 3 trials for each light source because of limitation on materials, but if I were to do this experiment again, I would perform at least 10 trials. This would reduce any possible errors. I want to finish with stating that studying the factors that affect plant growth are important for food production on Earth and in space. This knowledge has helped researchers determine which factors, including artificial light sources, are the best for growing plants in space stations. Thank you.